Hello, my name is Professor Nathan Lentz. I'm from John Jay College in New York City, and I'd like to tell you a little bit about my research. So I'm actually a forensic scientist, but I don't work too much on casework. I do research. So in academia, forensic science departments tend to be involved in method development and finding new ways to bring science into forensic investigations, criminal investigations. And that's really important because crime labs, if you see them on television shows and so on and so forth, they're pretty much fully dedicated to casework, to processing their backload of evidence. They don't have a lot of time or budget to do research, so the field wouldn't advance at all unless there was somebody looking for new ways and analyzing and critically looking at what we do and how we do it and pushing forward. So that's the role of academia. So academic forensic science is really involved in method development and analysis. So that's what my lab does. I have two areas of active research in this realm. One is I look at the post-mortem human microbiome. All right, so a lot of words to unpack there. Post-mortem means after death, and the microbiome is a collection of bacteria that live in, on, and around our body. I'm shooing away a fly, by the way, because another person shares this facility who is a forensic entomologist. So she studies insects and their role in crime scene investigations, usually also with post-mortem work. That's why we work together. So I look at the bacteria that specifically lives on human skin and how the communities of bacteria change after death, after the death of the human host. Because the reason why I think this is a useful thing to study in forensic science is because when you find a body out in the field, in an uncontrolled environment, the first question, in assuming this is likely a crime that's been committed, or at least it's possible, first thing investigators ask is how long has the person been there? How long have they been dead? And we don't actually have the best tools for that right now. We have some tools that work well in some environments, and we can kind of find upper limits and lower limits. That's basically based on insect colonization of the body, which is what my colleague studies here. But I thought that actually bacteria might be a very good indicator. So what we did was we collaborated with the Body Farm, which is a human decomposition facility in Tennessee. There are several of these in the United States now, but the oldest one, the one that we collaborated with, is actually at the University of Tennessee. So I went down there and we established collaboration. What we did was we looked at human decomposing bodies and we swabbed their skin in two places, the nose and the ear. We chose those very carefully for reasons I can talk about some other time. And we swabbed them for bacteria, brought the bacteria back to our lab, and then analyzed the communities of bacteria. This is a swab that we use to do that. It's kind of a specialized Q-tip. So it's a Q-tip, but it's inside of a tube. You see that there? So what I do is, we've been doing this with gloves, of course, but we pop open the tube, which protects the swab sample. And then you can extend out the swab, do it in the nose or the body of the decomposing person. You can only do this while they still have a nose and ear, which isn't forever. And then after you get the sample, you slide the tube back up, cover it to protect it, put it back in the sleeve, freeze it, and then we would take it back to the lab and analyze. And what we found in that project was that the bacteria do change in a very dramatic way following the death of the human host. There's a lot of bacteria that are there on our skin on a living person crash. Basically, you're looking at ecosystem collapse, and you lose most of those bacteria. But then new bacteria start to flourish, or bacteria that were there in low numbers start to flourish post-mortem. And then bacteria, of course, come in from the environment, probably brought by insects and other critters. And that's a dynamic community. It's constantly changing over several weeks of time. And what we were able to do is use computers, computer programs to find patterns in that data. Because we were looking at millions of data points over several points of time, we could actually determine, the computer could see the pattern of decomposition and the pattern of bacterial change during decomposition. And then we could use that to predict the decomposition time of a new sample, of an unknown sample. So we sort of trained the algorithm with known samples, and then we tested them on unknown. And we did this in an iterative process where we learned and got better and better at it. And we published that paper. We think it's a pretty good result. If you want to look up the results of this paper, it's in an open access journal called PLOS One. We specifically publish in open access whenever we can. It's more expensive for us, but I think it's very important to get the results of our research out to the public. So if you Google my name, Nathan Lentz, and PLOS One, or Nathan Lentz necrobiome, or even just microbiome will probably get you there. So you can find that paper on PLOS One and see what we did. A lot of computational work in that project, and I have a collaborator from the mathematics department that helps us. So that's a forensic science project that we've done and that we're pretty excited about. I think this is my lab, by the way, so you can see kind of a standard molecular biology setup here. There, over there is where we run all our gels and so forth. Here's our imaging. We have several kinds of imagers. Those are there's a SpeedVec and PCR machines. Standard molecular biology lab. There's a tissue culture suite over there through those doors, where we can grow cells if we need to. And it's a forensic science kind of operation, which is cool for me as a scientist because I don't get locked into one specific thing that I'm studying for years and years and years. I have projects, and each one of them takes me to a different area. So, for example, a project we're doing right now in the lab is on forensic botany. So we're actually looking at plants and how they could be of use in forensic investigation. The idea being is that there's plant residue all around us, especially in homes that have flowers in them. There's pollen. Pollen grains are very light, and there's millions of them, and they can attach to us. We go into our nostril and our clothes, and you can detect that later. So you might be able to actually place a person at a specific place by the pollen that they might have inhaled and got stuck in their nostrils. Even for days later, you can sometimes detect this pollen. So, but there aren't really good tools to do that now, at least not DNA-based tools. So we wanted to develop DNA barcodes, or actually develop the technology around DNA barcodes for use in forensic botany, in plant residue, that would be useful in a criminal investigation. So to do that, we've actually described some barcodes from 3 genes in a couple dozen species of very commonly found household plants, household flowering plants. Specifically, we wanted to focus on flowers for their tendency to produce pollen. So we did that. So we went to florist shops around New York City and bought a bunch of flowers that you commonly see, and we basically sequenced a few of their genes across species and looked for areas that were very different from one species to the next. We had to look at multiple genes because plant DNA sequences are very confusing and hard to decipher compared to some other species. So to find these barcodes was quite a challenge. But we looked at several genes, and we focused on a few of them. That work isn't published yet. We're still finishing it up, but I think we've got some tools now that forensic investigators might be able to use in the future to make use of plant residue. The next step, of course, would be to develop sort of real fingerprinting to connect evidence to a specific plant. A little bit down the road, but we have early ideas about how we're going to approach that using microsatellite DNA, similar to the STR microsatellite DNA that we use for human genetic testing, which we do some of that in our lab as well. Anyway, that's what forensic science research sort of looks like, at least for biologists. I'm a biologist, first and foremost, and it's been fun. You can look me up on Twitter or Tumblr or Facebook or email or whatever. My name is Nathan Lentz, Lents, and I work at John Jay College in New York City. All right, it's been fun talking to you. Bye-bye.